Jeopardy! In the studio today, we have the contestants Jean, Adenine, and Tim. So today's game is going to be focused around genetic editing and behavior in dogs. Each of our wonderful contestants have read the article Not Just a Hound Dog, an article by Bob Holmes to prepare for today's show. The categories for this interesting article are genome editing, looking to the future, and behavior in genes. Let's get started.
I'll take behavior in genes for 200.
According to research done by Alvarez, this gene is speculated to be linked to aggression in small yappy dog breeds.
What is IGF-1?
That is correct! Let's go to our prop master Cass for a more in-depth explanation on this gene.
In a study conducted by Carlos Alvarez, breeds like Dutchess and Yorkshire Terriers carry a version of at least one gene, IGF-1, that makes them more aggressive towards their owners and other familiar dogs. The gene is also partially responsible for their small size. Alvarez research shows that IGF-1 performance a range of physiological functions throughout the body and also codes for both size and aggression. It may do through different pathways.
Thank you, Cass. All right, now on to Tim.
I'll take, let's do genome editing for 100.
This was originally a defense mechanism in bacteria to cut up viral DNA, but is now used to toggle genes on and off, specifically and reliably in certain species.
What is CRISPR-Cas9?
Terrific! For those of you playing along at home, why don't we take a look at an example of how this process works. Once again, Cass.
Well, as we discussed in class, CRISPR is a set of 2 RNA segments, one of which is complementary to the target DNA. These 2 segments pair together and form a complex with Cas9, a nuclease enzyme that cuts target DNA in 2. When the CRISPR-Cas9 complex is formed, it moves to the target DNA. The complementary RNA CRISPR strand then bonds with the target DNA. Once this bond is successfully completed, Cas9 is signaled to cut the target DNA. This specified cutting strategy allows geneticists to create varying targets for the CRISPR-Cas9 system to any DNA sequence in many different organisms, allowing the turning on and off of genes.
Thank you. Now on to Jean.
I'll take behavior in genes for 300.
Alright, so according to a study comparing aggressive dogs to more mellow dogs conducted by Carlos Alvarez, there are several gene variants responsible for causing dogs to be more prone to aggressive behaviors than others. Of these genes, 2 are active in this part of the brain that helps to process fear.
What is the amygdala?
Correct! These contestants are truly astounding, ladies and gentlemen. And then back to you, Adenine.
I'll take looking to the future for 300.
So, studying genes and behavior in animals like dogs can help with understanding human tendencies as well. One study looked at how fast certain species of dog would turn to a human for help with a problem that the dog had. And it was found that several of these genes were related to behavior in humans with autism as well.
What is comparative genetics?
That is correct! As we discussed in class before, comparative genetics helps us to better understand genes across species by comparing one species to another.
I'll take looking into the future for 100.
Alright, so why could gene profiling be beneficial to the future of canine behavior?
What is creating better temperament?
According to the article by Combining Cognitive and Genetic Profiles, researchers plan to work out the genes behind intelligence, trainability, and sociability. The hopes of these projects can shine a light on how genes control traits we associate with personality.
Alright, on to you, Jean.
Okay, I'll take genome editing for 200.
Alright, when we look at certain species of dogs, for instance Dalmatians, we can see that these genes can cause common medical defects in those dogs. Many geneticists are hoping to use CRISPR and Cas9 technology to remove these from the genome.
That's a hard one. What are defective genes?
That is correct again.
I guess I'll take looking to the future for 200.
Alrighty. When we think about the future of genetics, there are things we'll be able to change in the genome that can help make ourselves and animals a lot healthier. However, we're unsure about what side effects might arise as multiple genes frequently code for one trait. For instance, removing the defective genes in Dalmatians could cause an unknown side effect. This is one of the reasons we aren't editing those genes now.
I'm not sure what the answer is here. This really is an ethical dilemma. What is an ethical dilemma?
That is correct. Our final question, Tim.
I'll go with behavior and genes for 100.
So according to the article, what is a significant signal of expression for certain genes?
What are breed characteristics?
Impressive. That
In several studies talked about in the article, they found that there were markers in canines' genomes associated with certain characteristics. For instance, there's a small section of DNA on chromosome 8 that is only represented in breeds that are pointer breeds. Another example would be specific markers they have found in herding breeds, which they could further associate, making them easier to train.
All right, so everybody give yourselves a round of applause. There is no one winner, because you're all winners, because you got to learn more about how behavior in canines is reflected in their genetics and vice versa.